Hello, my name is Trent Percy, and for my developmental biology project, I'm going to present on lung development and pulmonary cellular lineages. I'm going to give a very brief overview on how the germ cells incorporate into the different structures of the lungs, and then go over or take a molecular approach on some differentiation type stuff. So in the beginning, pretty much after you have a fertilized egg, it develops into a zygote, and then once it starts differentiating into its different tissue types, you get three different germ layers. So those three different germ layers are going to be the ectoderm, mesoderm, and the endoderm. Now the ectoderm, which is going to be represented by the blue label, is going to differentiate into basically two different types of tissue. You have your nervous tissue as well as your skin tissue. The mesoderm, represented in pink, is going to make up most of the connective tissue as well as the um, heart because it is a muscle tissue and as well as blood. Now the endoderm, represented in yellow there, is going to make up most of your internal organs. Now as far as the lung is concerned, two of these germ layers are going to make up the lung. That's going to be the lateral plate of the mesoderm as well as the endoderm. And the endoderm is going to make up the functional portion of the lungs, which are going to deal with gas exchange. So here's just an arrow that's showing how the lateral plate incorporates into the development of the lung. Now here it's a little bit of a repetition, but here we're going to show a diagram of how the three different germ layers are going to incorporate into the developed lung. So on the left here we have a pretty much an early embryo showing the three different germ layers. The first one, or the first layer I'm going to show, is the ectoderm, represented in blue here. That is not going to incorporate into the lung organ itself. The next one here is going to be this red layer here, and that's going to develop into the soft, um, or the excuse me, the smooth muscle that makes up the lung. The next is going to be the endoderm, of course, and that's going to make up the trachea, the bronchi, as well as the alveolar structures. So here I'm just representing pretty much the same thing that I just shown, that the endoderm is going to make up the trachea, bronchi, and alveoli, and then the mesoderm is going to make up the smooth muscle which are the lobes of the lung itself. So you can't really talk about developmental biology without talking about the fourth dimension, which is pretty much time. So as far as lung development is concerned, it's what's going to happen from around weeks 0 to 6. You're going to have basic organogenesis, to where you have the three germ layers that are differentiating into more functional tissue. And so that's going to happen between 0 and 6. Between weeks around 10 to 18 or 10 to 20, you're going to have actual development of the organ of what looks like lungs. Now, from weeks around 20 to 34 or so, you actually get cellular differentiation, to where the different cell types that make up the lung are differentiating into their functional units. Maybe they'll become sensing neuroendocrine cells, maybe they'll become like mucosal cells that produce mucus, or maybe they'll become like alveolar type cells that are going to actually deal with the gas exchange of the lung. And then, of course, you have birth, which happens at about 38 weeks post fertilization. Now here's a schematic that I had made for a publication that I contributed to, and this is going to go over pretty much the basic cellular differentiation of different lung cells, starting with the progenitor lung cell. Now, the progenitor lung cell is going to be SOX9, FOXA1/2, and NKX21 positive. Now, keep in mind NKX21, as that's going to play a role in our further discussions. Now, from the progenitor lung cell, that one cell is going to differentiate into two different cell types. Now, the two different cell types are going to be the proximal progenitor cell, which is represented in blue, or that dark blue there, and the distal or alveolar progenitor cell, represented in the dark green there. Now, as far as the nomenclature tells, the proximal cells are going to differentiate into the tracheal and bronchial tissues, while the distal cells are going to differentiate into the cells that make up the alveoli. Now, post differentiation of the proximal and distal cells, you get four main different cell types here. If we look to the left and you see the cells that differentiated from the proximal progenitor cell, you get a neuroendocrine cell. Those are going to be the cells that basically take messages and send messages. They're going to be ASCL1 and calca positive, and then you can also get the basal cell, which is TP63 and KRTS positive. Now, in the red text here is going to pretty much determine, or it's going to say what type of cancers these cells can give rise to. So the neuroendocrine cell can give rise to large and small cell carcinoma, and basal cells can give rise to squamous-like carcinomas. Now, if we go over to the right, as you can see, they are going to differentiate a little bit differently as far as their signals are concerned. So you can see that on the right you get SOX9 positivity, which that differs from the left. Also, after a little bit further differentiation, you also get surfactant positivity, which is going to be directly regulated by NKX2-1. So on the right here, we have two different alveolar type cells, which are going to, of course, make up the alveoli. So like I said before, let's keep NKX2-1 in mind. Now NKX2-1 is a master regulator in alveolar type 2 cells specifically. They are going to basically regulate target genes, for example, the surfactants. And surfactants are going to be necessary for gas exchange as well as maintaining the surface tension of the cells that are making contact with the air itself. So NKX2-1 is actually used as a biomarker for alveolar type 2 cells. It's something that pathologists use to diagnose lung adenocarcinoma. So it's going to be a very important protein for the cells to maintain in order for them to basically distinguish themselves as alveolar type 2 cells. Now, NKX2-1 is regulated in many different ways. So we can see on the periphery, which represents our cell membrane, we have a receptor pop-up. That's going to be the TGF-beta receptor. So TGF-beta plays a role in regulation of NKX2-1, and here is how. So once TGF-beta makes contact or activates the TGF-beta receptor, that complex there is going to activate by phosphorylation SMAD3. Now, SMAD3 itself is a repressor of NKX2-1. Now, similarly, another mechanism is going to pretty much do the same thing, and that's going to be hedgehog signaling or sonic hedgehog signaling. So typically, when the pathway is not stimulated, the patch protein that exists on the cell surface is going to inhibit the smoothen protein. Now, if you have activation by the sonic hedgehog protein or ligand, that is going to interrupt that signal to where the smoothen protein can now migrate to the cell surface. Now that can go ahead and cascade down its signal. So that is going to activate a protein called GLE2 and GLE3 or one or the other. That is going to transcriptionally activate a protein or a gene encoding protein called FOXA1 and FOXA2, and those are going to repress NKX2-1. Now, if you could scroll back and look back at my schematic, we could see that FOXA2 positivity is pretty much consistent with the proximal cells, or the cells that don't differentiate into the alveolar-like cells. Now, that's pretty much what I have on repression of NKX2-1, but what about activation? Now, activation, one of the ways, now there's a few different ways, but one of the ways is going to be through JAK-STAT signaling. So the JAK receptor or associated JAK-associated receptor is going to be found on the cellular membrane and can be activated by a molecule such as IL-6. Now, once something like IL-6 activates this receptor, basically JAK1 and JAK2 associated receptors are going to dimerize. Once they dimerize, they're going to recruit STAT3. Then STAT3 can go ahead and dimerize itself, be activated by this receptor by phosphorylation, and then STAT3 itself is a positive regulator of NKX2-1. So all these things considered, these are a bunch of different important processes when it comes to lung cell differentiation. You can also see that many of these different pathways are going to also regulate a lot of different tissue types. So that is my presentation, and I thank you for listening.